All right. Hi, guys. Apologies again for the technical issues for today's lecture. For those of you where the audio was interrupted constantly, I did mention that it did not seem to be worthwhile to try and continue forcing it, but that I would record what I wanted to share for today's lecture, and I'll upload this video for you to YouTube, and so you can see it there. And then please feel free to ask any questions via the Canvas chat or via email before Friday's lecture. And hopefully by Friday, this will have been resolved and we have better connectivity and can have a proper class discussion. So for now, option 2, I'm pre-recording here what I wanted to share with you today, which is largely course logistics, and so starting to dive into specialized metabolism. Right? And again, if you couldn't hear me today, usually for this class, I don't have dedicated office hours. I really have usually an open door policy, especially since we're working with discussions and you guys are working on proposals. I'm happy to work with you directly. So if you want some more guidance outside of the classroom, just shoot me an email and we can set up a Zoom meeting or just a phone call and discuss those things, all right? Okay, so let's dive in. Mostly some of the course logistics. So what I want to you to get out of this class really is to gain an understanding of the biochemical and the genetic logic of plant specialized metabolism. So really looking at the major processes of how plant natural products are formed, their chemical diversity, their functional diversity, and how this relates to plant stress adaptation, for example, but also to a variety of industrial applications. And sort of building on this basic foundation of plant natural product, biochemistry and physiology, I usually put a lot of emphasis of this course on the toolbox. So talking about the variety of experimental approaches that are out there for the analysis and the engineering of specialized metabolism, and specifically how you can combine these different kind of tools to ask different kind of questions that you may have as part of your current projects. In the past, I've used sort of this combining methods to look at strategies for metabolic engineering applications in plants and microbial hosts. We can do the same here, or we can change emphasis a little bit based on class interest. And so after that, what I end on in the class is to sort of take what we've learned about ecological relevance, industrial relevance, possibilities for applications, and how this relates to aspects of bioethics, commercialization, and intellectual property, specifically surrounding issues of plant biotech. And so here's our lecture schedule. As I had mentioned in blue, these are more the lecture type days where I want to give you an introduction and overview of the background on plant natural products, as well as experimental approaches that we then can use for the individual discussion sessions. And so they are highlighted here in yellow, where I really hope, technology provided, that we can have a proper discussion around these topics. And then in brown, we have 3 sort of guest speakers. Nitsan Shabak and Somin Nandi will actually give guest lectures on protein structure biology and chemical and biological engineering, respectively. And then next week, we actually have a library session with Leila Kabugos, where we will just practice and some of the resources available for literary research, including, but also going beyond Google Scholar, as well as tools for auto-editing references, reference libraries, and so forth. I've done this last year, and the students found it quite valuable, so I thought I'll do it again. And I hope this will work in a remote session, but Leila is putting something together over Zoom. All right. Then to sort of the exam part of the course, it will be fairly minor. There will be one midterm, which is a take-home exam. So it will be based on an article and sort of asking you to summarize it and to provide your opinion on a couple of questions around this article. It'll be open as a Canvas quiz, ideally, or I just have you send me Word files. I'll let you know closer to the time. I'm aiming for putting this out mid-quarter, so around week 5, and it will be open probably for a few days or even a week. So I'm happy to be really flexible there this year, depending on how this conflicts with some other exams you might be having. But again, technology provided a big part for me for this course is that we have a discussion around these topics. So your participation in these discussions is really important to me. And then finally, we don't have a final exam, but a final essentially is a 2-page research proposal. I have provided some guidelines as well as an example of a proposal already in Canvas, so you can look at that. In past years, people have used this to actually prepare for example, the NSF proposals or other thesis proposals, your QE proposal, for example. So I'm more than happy for you to do that. And it's just then also an opportunity for you to practice and get feedback from myself as well from your peers in the class on your proposal work and just to improve. It's something that we will all do continuously throughout our career. So I always feel this is a more meaningful approach than simply a testing exam for a graduate class. So I hope this will work for everyone. All right. To the discussion sessions, most of you already have signed up. So those people who haven't signed up yet, there is only sort of one topic left here. So please sign up for that. And because of our class size this year, it's a lot larger than the classes I had in the past years. We have a few more people for each discussion, but I think there's enough to do for everyone to contribute. And so what I would hope to do is actually deviate from sort of the traditional discussion format that you might be more accustomed to, where basically explaining a figure is being distributed among individual people, and we mostly have a discussion based on these figures. I feel like at the graduate level, we should move beyond that and really talk about these articles in the context of their contribution to the broader field, and really start thinking about the knowledge that is being put out there and what it means to the development of more knowledge, to development of new approaches and so forth. So I really hope this to be a lively and broader discussion. So the way I usually set this up is that everyone reads the article and then except for the lead team that will lead the discussion, fill out the discussion form that I have uploaded on Canvas already. And then the lead team, so those 3 people would provide a synopsis of the article. So really looking at what are the major conclusions, what are the major take-home messages from this article. For example, what are the strengths and weaknesses that you see in this article? And really sort of give an assessment, much like a review of the article. And then we would open it up for questions and a discussion around the article itself. And the lead team, and obviously there to help, would moderate that discussion. And in addition to that, but after that discussion more specific to the article, what I would really like to do is that the lead team prepare a few questions sort of pertaining to what the article contributes to the field and moderate that discussion. So just to give you an idea of what I mean by that is, for example, for our first discussion next week, the article that I suggested is this one highlighted here by Ruben Peters' group. So this is focusing on the evolution and diversification of diterpenoid metabolism. And so based on this article, there are a few questions that I could think of, but you can obviously come up with others, and I'm happy to help you define these questions. You could ask, based on this article, how does the knowledge of specialized metabolism and its diversification that is highlighted in this article change our understanding of, for example, plant speciation, environmental adaptation, and so forth? Another question could be, how can this knowledge be utilized in industry, right? How can we learn something from this for developing engineering tools? And also, based on the current knowledge of this article, this is this review article, is what are the next critical steps, right? What is missing? What are the big questions in the field that need to be addressed? So I'm really thinking about broader type questions here that these articles that I've chosen open up to have a discussion and really think about this field more broadly. That said, what I would recommend to facilitate a good discussion is that the lead teams work with me so that we can post these broader questions maybe 2 days before our discussion time, so that everyone in the class has time to look at them, does their own literature research or form an opinion, and so we can have a discussion that is a bit more informed and everybody had enough time to prepare for the discussion rounds. So I hope that will work for everyone and we have a good discussion. It has worked well in the past, and so I would like to continue this model. That said, the articles I've chosen, these are recommendations. If you are really keen on another article on the topic of the discussion session that you've chosen that you would really like to discuss, please let me know and I'm happy to entertain that option. All right, so that's for the discussions. Now to the proposal. I already mentioned it here, even though it's basically our final, because I would encourage you guys to start early on this. The earlier you start, the more time we have to work together on really fine-tuning your proposal and work through the troubleshooting and so forth. Generally, what I would hope to see is basically a 2-page white paper style proposal. So a white paper usually means it's a broad approach, the feasibility, the need outlined rather than specific details. So I do not expect you, for example, for the experimental approaches to really lay out all these experiments in detail. Right? What I want to see is, this is what I propose, this is why it's important, this is why I should do it, I have all the resources to do this is why it's important to the field, and this is how we're going to do it, and this is why this approach is the most feasible one. Okay? So try to imagine this is going to a funding agency or this is going to a decision board for ag, biotech, or other biotech industry. So you have full freedom. If you want to make a proposal that is proposing a new company, that's fine. If it's a proposal that's geared toward foundational research and the relevant agencies, that's also fine. So that's up to you, whatever is of most interest to you. So outlined here, and I have this proposal guide also on Canvas. I just want to give you a few pointers on how I would structure it. So you should start with sort of a good, catchy title. That's always good. Right? Not too long, not too short, but it should be descriptive. Then you should talk about the project significance, right? Like, think about writing a proposal and keep in mind that the people who review it are probably busy people. So unless you convince these people right off the gate, it always is hard to get a proposal funded. I can assure you about this from my experience. So really think about this, a brief product summary and how you can convince that this is important and to make people continue be interested in reading the rest of the proposal. So really think about, this is the moment where you convince, for example, an agency or a venture capital group to give you money. Right? So what you should do here is to state the research problem, state the major goals, but very briefly. And then more focus on what is the main issue that is being addressed in the broader context. Why is it important? Why now? And how can we do this? I convince that agency that you have the tools to do it. That then is followed by the research plan. You should have provided enough sufficient background information for people to understand the project, but without going into all the details that are only relevant to you or to other experts. What I would highlight is, what are the knowledge gaps, right? Why is this important? What are technical hurdles if you're more looking into a tool development proposal? Why is your research uniquely suitable to address this challenge? Right? That might be a bit challenging for you because you're early in your career, but think about, for example, the resources that you have at UC Davis or in your lab that you could bring forward as a strength here. Describe the experimental strategy to achieve those goals, but again, do so briefly and focus only on the critical aspects. Right? Why is this the best approach, right? And also, what is your plan B? What do you do if this plan fails, right? And then you follow that research plan with your broader impacts, where you really talk about not just a copy of the project description, but really focusing on what are the benefits of the project to those in the community or those who are funding that work, right? For example, how would this contribute to enable further innovation, right? How is this knowledge relevant to the broader community? In some cases, you don't have to necessarily focus on it here, but if what you're proposing could have some ethical consequences, you should address those, right? And then literature cited should only be the major ones, right? And very briefly, but they should be included. And in this case, they have to be part of the 2 pages. So I hope this is helpful. Again, look up the example I have on Canvas and the additional guidelines. And obviously, we can talk more about this throughout the quarter. All right, so that's what I have for course logistics. Now I just want to sort of dive into some introductions to specialized metabolism and get us going. And so this is a part that usually would be very interactive, and I really would usually work with you on sort of developing some concepts and so forth. So what I've done here is just show a blank page, because really I want you to essentially just pause the video here and think for yourself about some of the concepts that you could think about in some of these topics that I'm addressing. This first one, what I want to do is to just compare general and specialized primary and secondary metabolism. And just for the record, primary and secondary is sort of the traditional way of calling it. It's been increasingly changed over the last years from general to specialized metabolism, because people are increasingly recognizing how important specialized metabolites are. So the community felt increasingly that secondary wasn't an accurate description for this entire group of metabolites. But what I want you to do essentially here is, if you want to just stop the video and think about how would you define general and specialized metabolism, and maybe think about a few aspects that you think are unique to general or specialized metabolism and how those two compare. All right, so if you have paused, you may have something written down now. If not, you can just follow me and look at the concepts that I am considering for this comparison of general and specialized metabolites and metabolism. So this is how would I define it. But general metabolites are those that are really conserved across the plant kingdom, and they typically are defined as having essential functions that are specifically designated to nutrition, growth, and development. So the general metabolites that really sustain life for an organism independent of its environmental conditions. And so this would include your amino acids, that will include your sugars, and it would include your hormones, for example. So this is what we would definitely consider general metabolites. And I should say that in general, there's a lot of gray zone between those two. Right? Oftentimes, a specialized metabolite also touches general metabolism and vice versa. But in general, comparing to these really critical foundational metabolites, what would define as specialized metabolites or natural products is that they're typically not conserved across the plant kingdom. They're typically taxonomically restricted, so they may occur in only individual plant families or even genera and individual species. We even see differences between individual plants within a species. And in addition to that, their major role really is to mediate communication of plants with their environment. That can be cooperative or defensive, but this really is sort of the key to many, many specialized metabolites, that their role really is environment dependent and really mediating that communication. As part of this, as languages basically evolve and diversify, unlike general metabolism, specialized metabolism is extremely diverse and actually is the fastest evolving aspect of plant metabolism, right? Basically in the arms race of plants with their environment and their communication, they continue to evolve new compounds to help fine-tune that interaction with the environment. But seeing this sort of major difference, this gray zone I mentioned between functions is also really reflected in structures. So you cannot say that this metabolite group is a specialized metabolite group just defined on its structure. They're really fine differences. And sort of one example I can highlight here is two precursors of diterpene metabolism. The one here being end copolyl diphosphate, and so end here highlights the enantiomer here at C9 and C10, which is facing backwards here. And so its close sibling here plus copolyl diphosphate, so it's enantiomer with the opposite stereochemistry here at C9 and 10. Those are obviously structurally very similar, but this difference in stereochemistry is actually enough to completely change the bioactivity of these compounds in their relationship to general and specialized metabolism. So N-copolyl diphosphate is actually a precursor for production of ghrelin, so plant growth hormones. And plus copolyl diphosphate is actually one of the most common precursors to bioactive diterpenes that are function in pathogen defense and insect defense. So this very subtle difference in stereochemistry has actually profound impact on how plants are using these compounds and the bioactivity of these compounds in different ways. All right, so that we have a bit of an understanding of the general comparison of general and specialized metabolites, I want to first focus a bit on what are the specialized metabolite classes. And sort of those among you who have been in the plant biology core class 200B have seen some of this before, but I'm pretty sure it's probably a good reminder. You probably don't remember all of that very quick and short week we had to talk about metabolism. So the first thing I want to do is to sort of summarize for us some of the major specialized metabolite classes and see what actually is out there in the plant kingdom. And so again, I would just encourage you to stop the video here and maybe pin down for yourself what classes you're aware of, what you know in terms of specialized metabolites that you can think of, and then we can move on together and see how many of those that are actually present in plants you could cover. All right, just assume you did that, so let's move on. And so this is a list. It's not fully comprehensive. There are more specialized metabolites that are much more rare. So I'm focusing here on the major classes. The largest one of the typically oxygenated hydrocarbon compounds, and those that are not nitrogenous, are the terpenoids or terpenes. They are actually the largest and most diverse class of plant natural products and the most widely distributed in terms of how many plant families, plant species are producing these compounds. They are divided up into a number of subgroups that are largely divided by the number of carbons in the backbone of terpenoids. And so we have the hemiterpenoids that are 5 carbon compounds, then mono is carbons 10, di is 20 carbons, triterpenoids are 30 carbons. Your carotenoids are 40 carbons, and then rubber is actually a polymer of terpene units. The next biggest one then, and the next most diverse, are the phenylpropanoids. Here they're also extremely widely distributed, a little smaller in terms of numbers. Terpenes, we are looking at probably more than 100,000 different compounds. Phenylpropanoids, if you look into the most recent literature, it's probably more around 20 to 30,000 compounds. But still very, very diverse, very widely distributed across the plant kingdom. And so there again, there are numerous subgroups. I'm only mentioning a few here. Small phenolic acids are very common. Stilbenes are very common. Lignin and lignanes. So lignin is actually an interesting example where you could see it as a general metabolite because it provides structure for a plant. but it also provides structural defense against, for example, boring insects or other herbivores. And so it can also be seen as a defensive metabolite and therefore a specialized metabolite. And then we have a number of flavonoids. So these are specialized metabolites with a lot of different functions. And again, they are subdivided into different ketones and alcohols and so forth. We'll see some of those in a bit more detail when we talk about the natural product classes separately in separate lectures. I actually like to include the waxes here that are often sort of not really considered much, but if you think about the chemical diversity of waxes on plant surfaces and how they contribute to plant defense as a structural defense, they're actually also quite important. And over the last few years, research really has shown that the diversity of wax structures is much larger than originally anticipated. And so there's actually more research happening now to look into the biosynthesis and diversity of waxes. Then on the other side of that world of the hydrocarbons, we have this group of nitrogen-containing compounds. And so probably the most famous one are your alkaloids, probably something around 10, maybe 15,000 compounds. They are actually more restricted than the terpenoids and phenylpropanoids, as well as the waxes. They are really limited to the to angiosperms. As far as I know, no alkaloids have yet been found in gymnosperm or other related species. So they are just evolved more recently. They are widely distributed across the angiosperm lineage, and again, they are broken down into a large variety of subgroups. Some of the more common ones are the benzyl isoquinoline alkaloids, as well as the tropane and indole alkaloids. We will look very briefly into their biosynthesis. It's extremely complex and species specific. So we'll just have a look at one or two examples. Then moving forward, glucosinolates are a very important group, much smaller, a few thousand compounds, if that. And also much more restricted. There are, as far as I know, so far restricted to the order of brassicales. So there are a very lineage-specific evolution of specialized metabolites, but in these species are heavily used. And I know some of you have a project interest in glucosinolates, so we will actually be looking at those in a little bit more detail throughout the quarter. Actually a bit similar in their structure and in their physiological role are the cyanogenic glycosides. Again, a smaller group, limited to the angiosperms, but more widely distributed than the glucosinolates. So there are a number of less related angiosperm species that are producing cyanogenic glycosides. And then they have a few more here that are less common, but nonetheless important. NPAAs, or non-protein amino acids, actually occur in a number of angiosperm species. And those are things like GABA and L-DOPA that are, for example, used as specialized metabolites in legumes and grasses and some other species. Amides and alkamides are their own subgroup, but there isn't actually all that much research out there on these subgroups, so it's not as well understood how diverse they are in comparison to some of these other natural product classes. There is some recent work on echinacea, and so alkamides are actually one of the major bioactive natural product classes that are relevant for the therapeutic effects of chamomile and other species of the echinacea. And then last but not least on the list here are the polyketides. They are abundant in select species, a kind of a unique, really diverse group, very diverse biosynthetic pathways. In plants, you find them in several species. They are more common actually in fungi, and they produce a lot of these polyketides. All right, so our focus will be on the terpenoids, phenylpropanoids, alkaloids, glucosinolates, and cyanogenic glycosides for this quarter. Unless some of you have a particular interest in these others, I would say in the interest of time, we'll focus on these major ones. All right, so now that we have an overview of the natural product classes that are out there, let's look a little bit into their physiological roles. And again, we'll focus on some of these in more detail when we touch on the individual natural product classes. But I want to have a look here more broadly at what are the general functional roles of these compounds. So before we do that, I think it will be important to think about a bit what actually makes these compounds bioactive. And essentially, there are two contributing factors, chemically speaking, that contribute to the bioactivity of these chemicals. And so the two are essentially the three-dimensional coordination and configuration of these compounds, which really impacts how they can interact, especially with other enzymes, which has a lot to do with their bioactivity. And the second one being functional modifications. For the first aspect of the configuration of the molecule, the first aspect we look at is hybridization. What you usually see in any natural product is as you're moving from the linear sp hybridized molecules to the planar sp2 molecules to the more common sp3 tetrahedral arrangements of hybridization, you actually see an increase in bioactivity. And that makes a lot of sense because the sp3 hybridized carbon atoms really give it diversity. They allow for stereoisomers. They allow for variation in configuration of the larger molecules that allow for differences in interacting with other molecules, specifically with enzymes. Of those molecules where we actually understand the mechanism of action, we'll see that bioactivity really has a lot to do with interfering or modifying the activity of other enzymes. Right, then following up on the hybridization, this is an additional figure actually now showing different bioactive compounds. And so there are multiple here. You have nitrogen-containing compounds here that are very toxic compounds. You have centellol here, which is a terpene volatile that is likely used in plant communication, but is mostly used in the fragrance industry. We have stevioside here. Many people will know this among you, right? This is a natural sweetener. It's a diterpene that is glycosylated. And here we have a larger triterpenoid, evanescin, which is a very strong anti-fedant agent and also produced in a number of species. But what you see here is that we have highlighted here all these chiral centers. All of these are the carbons that have sp3 type hybridization. That allows them to form stereoisomers. That allows a molecule to generate more new chiral centers. And I highlight these here because these are attached already to functional modifications. And so here, right, we're generating an sp3 hybridized carbon through this functional modification, the hydroxylation. So the ratio of these sp3 carbons over the total number of carbons can actually be used as a relative measure of potential bioactivity of natural products. So that's important to keep in mind, right? So using chirality, using chiral centers for modification and structural diversity really has been a major contributing factor to the functional diversity of these compounds. And so then building on the chirality of these compounds and the potential for chiral centers, probably the major contributor to bioactivity is functional modification, right? And you see a lot of these compounds are functionally modified. A lot of these are oxygenation, lactone ring formations, simple hydroxylations, amine group formations, attachment of sulfuryl groups, and so forth, right? So on top of these hydrocarbon backbones and the chirality of these different hydrocarbon backbones, the functional modification of these is likely the major driver of bioactivity and potency of bioactivity, but also diversity of bioactivity. So keep this in mind from a chemical perspective, what actually is making these compounds bioactive. All right, so now that we have sort of thought about this a little bit, I want to pause the video just for a moment again here, or you can pause it. So this would usually be a spot again where we would have more of a group discussion and thinking about the list of major natural products that we had identified now and that are listed here on the left already, to think about some of the physiological functions that you can think of these compounds that you may have touched on in your studies before. So just take a few minutes and just jot down a few physiological functions that you can think of that these compounds have. All right, so let's do this together. So I listed a few things here, and you can sort of largely divide biological functions for this entire group of natural products between cooperative functions and defensive functions. So under the cooperative ones, you can definitely think of pollinator attraction as well as tritrophic interactions. So these are interactions where, for example, a plant can be attacked by a caterpillar and would use volatile compounds to attract, for example, a predatory wasp that is a natural predator of this caterpillar. So it's sort of a three-way interaction of a plant to defend itself by attracting the natural enemy of the predator that is attacking the plant. And so both pollinator attraction and these tritrophic interaction usually will include more volatile type of compounds that can be communication signals over a longer distance, right? And can be volatile enough to actually be transferred through the air. Then seed disperser attraction, these are all your pigments, your colors, right? And then those are the major three, but more recently, in recent years, there are actually more and more articles coming out that highlight the function of small molecules that can be released into the soil, into the rhizosphere, and can mediate interactions with the microbiome. And also there are more and more examples and suggestions of metabolites that actually work in systemic signaling, so transfer of defense signals, particularly throughout the plant, rather than having them as local defenses at the point of attack only. Okay. And these can be cooperative or defensive, so I probably should have listed them already down here under defensive functions. But then some of the major defenses, these are herbivore deterrents, so things that are bitter, that have anti-feedant functions. Many of these compounds are involved in microbial pathogen defenses. They can be antifungal, they can be antibacterial, they can be both. And in some cases, we also have seen antiviral activities. And then again, plant-plant signaling can be involved here. So similar to our pollinator attraction up here, plant-plant signaling can also be defensive. So other plants can actually recognize volatile defense compounds that are emitted from a neighboring plant and can actually activate their own internal defenses, essentially as a priming for possible defense in response to recognizing that another plant is sending out defensive signals. So there are some really fascinating examples with sagebrush and tobacco out there. Then are some others here defensive. There's UV protection. Some of the metabolite groups are functioning in that area. And we also see examples now where we find significant accumulation of root-specific metabolites that accumulate in response to drought. And so there's quite a few hypotheses out there that this also may be in conjunction with microbiome interactions, right? So there's a lot of more research going on, especially in below ground plant environment interactions that are far less understood as compared, for example, to the pathogen type defense mechanisms. So what I want to do next is sort of work through with you and look at some of the metabolite classes that we highlighted here, and maybe just write down some of the specific functions of these individual metabolites. So again, this would be usually what we would do interactively. So this video is certainly not the best option, but let's try. And so let's start with maybe some of the more narrowly distributed metabolite classes, like the glucosinolates, for example. Right, these, right, for those of you who work on them, are specifically involved in herbivore deterrence. So number 6 would be relevant here. They also have some other functions. They can actually also serve as in nutrition. We'll actually see some examples of both cyanogenic glycosides and glucosinolates that they can vary in function somewhat. But the major part will be actually herbivore defense. Right? So the list I'm putting together here may not be fully inclusive, but I want to highlight the major functions of these compounds. Then similarly, right, cyanogenic glycosides also, these are your cyanogenic bombs. They actually also are involved in herbivore deterrence in these species. Right? Alkaloids, actually not all that much is known. We do know that nicotine, for example, as one of the major alkaloids, is very effective in herbivore deterrence against tobacco-specific caterpillars. So again, number 6 here would be relevant. But there is little research actually on the physiological relevance of alkaloids. Really, the vast majority of research on alkaloids is in the therapeutic and medicinal areas. But for the most part, we can assume that these compounds, their major role will be herbivore defense. Several of these may also well have or do have, to some degree, microbial pathogen defenses, but this would be a secondary role here to the herbivore defense. Then your phenylpropanoids, a much more diverse metabolite class, right? Much more widely distributed. And this really also lends itself clearly to a more versatile type of functions here. So some of these, like phenolic acids, certainly can be herbivore defenses if they're bitter. They also certainly are involved in microbial defenses. Some small phenolic acids may actually be volatile enough to be involved in plant-plant signaling, and so forth. Right? And then on the cooperative side, they're probably more defensive in this particular case, but they could be involved also in some microbiome interactions. There's some work going on that. Tannins are really much more structural defenses. So again, number 6 here, they're feeding deterrence. They also probably have some microbial pathogen defenses. There are some reports out there on that. Flavonoids are really versatile. They're pigments, right? They're actually colored. So number 1 would be a factor here. Number 3 would be a factor here. So really aiding in seed dispersal and pollinator attraction. They also, some of the flavonoids actually have been shown to mediate interaction with root-dwelling microbes and soil-dwelling microbes. So number 4 is a factor here. Flavonoids probably have less impact here in herbivore deterrence. They may have some microbial pathogen defense functions. So I list those here in brackets, but I think that's not a major role here. They're certainly involved in UV protection also due to the arrangement of double bonds. And I would say because of their role in microbiome interactions, probably also in TAN. But there isn't much out there, but I would put that hypothesis out there that they're likely also involved to some degree in drought and other abiotic stress defenses. And then as a subgroup, we can sort of list the anthocyanins under the flavonoids, obviously, but they are the colored ones, so there would definitely be under 3 here, but also in UV protection, these would be some major roles here. And then to the terpenoids, even more diverse in structure, even more diverse in function and distribution. So they cover almost everything, in part because of the large range of structural diversity, all the way from really small volatile compounds to large, complex, heavily functionalized type of non-volatile compounds. Certainly the mono and sesquiterpenes are the smaller, more volatile compounds. They would be involved in pollinator, in pollinator attraction. They're certainly involved in tritrophic interaction. They're examples of that. There are examples of them being involved in microbiome interactions. What else do we have? Terpenates outside, most of these compounds are less involved, most likely in herbivore deterrence, but they are to some degree less so the volatile compounds though. We do have many examples of them being involved in plant-plant signaling. Their lack of conjugated double bonds and aromatic rings, they are less involved in UV protection, but there are some studies that at least suggest a role in drought tolerance, for example. Again, I put it in brackets because evidence for this is still pretty scarce. The diterpenoids, most of them don't smell all that good. They are more involved in defensive functions outside of the hormones. So these are involved a lot in defense. Some show roles in herbivore defense, but the vast majority of these compounds is involved in pathogen defense. I'm actually going to underline this here. There's less known in plant-plant signaling, but they actually can have allelopathic functions, so suppressing the growth of neighboring plants. So they actually can do that, but in a defensive fashion. Again, barely any conjugated double bonds, so less involved in UV. But there's actually some evidence, in part actually from our own group, that they're involved in drought tolerance. We just don't exactly know how that works. Then the triterpenoids, they're a bit different. So we're getting heavier and heavier in terms of lack of volatility and overall size and functional modification of these compounds. Triterpenoids tend to be really bitter and really toxic. So their major function really is herbivore defenses and microbial pathogen defenses. There might be some examples here also in terms of microbiome interactions, but it's less clear. And then last but not least, the carotenoids, so the 40-carbon terpenoids, they're colored. They actually also have a clear role in general metabolism as part of the photosynthesis and the photosynthetic systems. As it relates to specialized metabolism, they are certainly involved in UV protection due to their conjugated double bond arrangements, and they're also certainly involved in seed disperser attraction due to their bright reddish, yellowish type of colors. Right? So you can see there's a lot of diversity in the functions, and there's a clear relationship of the chemical diversity and the range of distribution of these compounds to the versatile and the diversity of physiological functions that we actually see for these types of compounds. Right? Whereas those that are more family specific tend to be more defensive functions, and especially those that we are touching on this quarter seem to be more involved in general herbivore defenses. Right? For the glucosinolates, though, we should definitely also add 7 here. I've overlooked that. They certainly also have clear pathogen defensive functions. All right, so I hope I didn't overlook everything. If I did, post it to me and we'll look into it. But this is sort of what I wanted to cover here as a broader overview. All right. So again, this is just to summarize this. On the cooperative side, pollinator attraction interaction, seed disperser interaction, other beneficial cooperative type interactions are really important. The majority of compounds that are involved in this, outside of the pigments, they're larger, but for the most part, these are your pigments, carotenoids, anthocyanins, other flavonoids. The betalanes are your pigments, but then all other kinds of interactions that are not pigments are typically volatile organic compounds, so your VOCs. And this includes terpenoids, green leaf volatiles. These are often small alcohols. There can be small phenolics are involved in this group too. And other small molecule phenylpropanoids that really cover this group of of interactions and physiological functions. And so one example here, for example, in the pigments is your anthocyanins. So if you actually have a look here at all these common structures, right, so you can tell all these compounds have essentially the same structural backbone. This is a typical anthocyanin flavonoid type backbone. But then you see you have slightly different double bond arrangements. Right, there are additional hydroxy groups or other functional groups, different positions and numbers of hydroxy groups. And so those differences in hydroxylation and double bond arrangements really make the color of anthocyanidins all the way from sort of a bright light orange to a dark purple, almost blue. And so these have major roles in the development, especially of flower colors, but also fruit colors. And the extraction of seed dispersers and pollinators. And usually in class for the Vox, I have a few examples that you can smell. That obviously doesn't work right now, but these are usually the fragrant volatile compounds that then pollinators can be attracted to and can recognize. Then on the defensive side of things, we really compare two major differences. The one would be the structural defenses. So these would be your resins. Lignins would fall into that category. Waxes would fall into this category. So essentially all the specialized metabolites that form physical barriers to pathogens, to herbivores, and provide a deterrence that is structural in nature. And so that we compare to chemical defenses. This is certainly the most abundant physiological functions of specialized metabolites is in the realm of chemical defenses. And so what you compare here again, by definition, is phytoalexins to phytoanticipins, where phytoalexins are essentially bioactive antimicrobial compounds that are inducible in response to stress. And phytoanticipins also have antimicrobial functions, but tend to be constantly produced and constantly present in plant tissue and don't show stress-inducible accumulation patterns. So that's sort of the major difference between those two groups. And these really cover largely all of the groups we touched on. So the alkaloids, phenylpropanoids, especially the higher molecular weight ones, terpenoids, glucosinolates, sanguinic glycosides, all of these are sort of the major groups of constitutive or inducible chemical defenses. All right, so this covers sort of the broad overview of physiological roles. I will look at sort of specific function and specific examples as we're starting to touch on these individual metabolite groups. So we'll see this in the next lectures too. I want to now move on to the principles of biosynthesis and look at where are these things produced, and then how are they produced. So again, you can pause here and just sort of think for yourself a little bit of any kind of plant tissues or organelles, suborganelles that you can think of that you would consider are sort of specifically dedicated to the production of specialized metabolites. And so to just test of what you found, let's go through it together. So there's actually a number of of tissues and organs where specialized metabolites are produced. And a major reason for this is that, right, we had just talked about a lot of these compounds being toxic, being phytotoxins. And so you want to prevent two things. You want to prevent autotoxicity in this case. So it's beneficial in many cases to store these compounds away from the cell environment in specific organs or suborganelles to prevent that autotoxicity. And in addition, you want them in an area where they actually are beneficial to the defense. And so one of these tissues or organs are trichomes. So these are essentially small multicellular appendages of the epidermis. And so what you have here is that you basically have a number of epidermal cells here that are forming this stipple here, where you basically have a small branch coming off the epidermis. And then on the top here, you have a secretory vesicle sitting on top of the trichome. And in this, what you have is these stipe cells that are connectors to the bases, and you have these secretory cells here. And so these secretory cells actually are where a lot of the specialized metabolite biosynthesis in trichomes is occurring, and then they're being secreted into this vesicle. And so this allows the plant to do exactly these two things. It prevents ototoxicity because you're generating these compounds away from the major leaf cells in the specific organ. And it is forming a barrier of defense because you can easily imagine, right, if the herbivore attacks this plant, you can see all these trichomes here on the stem of this plant here, looks like a tomato. But it will actually touch these trichomes, and then this secretory vesicle would break off and will release the toxic compounds. Right? So it's a very important one that you find in many species, especially Lamiaceae, Asteraceae, Solanaceae species are very common to produce these trichomes. Right? And then there are different kinds. There are glandular and non-glandular trichomes, so they don't always have to look like this. Some actually may have formed different structures. Some are much more bulby here. This depends in part on the plant species and in part on the specific functions of these trichomes. Another one that we find that is more specific to gymnosperms and especially coniferous species, needle trees, follows the same principle, but using it slightly differently. Here we see resin ducts, right? And so what you see here in this cross section of a coniferous stem is that you see all these ducts here sitting right outside the cambial zone in the bark tissue. And if you actually zoom in here, this is a stained zoom in of this cross section. You have it stained, you can actually see all the lipid compounds here. And what you see, this is a resin duct here. And so on the inside here, you have these epithelial cells. So they're very much like the secretory cells in the trichomes I just showed you, same function. These guys are actually producing the metabolites, and then they're secreted into the resin duct. The end result is, right, if you tap the tree, you have huge amounts of this resin coming out. And so this resin is probably 98% terpenes for most of these coniferous species, and it has a toxic function. It is toxic to especially fungal pathogens, but also to stem-boring insects like the bark beetle here. And in addition to its toxicity, these guys, as you have seen if you walk through the woods, right, they polymerize and will actually trap, for example, the insect that is trying to enter the tree, and they will also seal this wound that the insect has created to prevent further attack by fungal pathogens. So they're both structural and chemical defenses. Very similar to these resin ducts are leticifers. These are actually specific to angiosperms, and we find them particularly in opium poppy as sort of an infamous example, but also in species of the Euphorbiaceae, for example. So everything, if you sort of rip a leaf apart and you see this white latexy milk coming out, these usually come from leticifers. And so the strategy here is very similar to the resin ducts, that you can have sort of epithelial cells lying along what is for a leticifer right here. Again, there are different kinds that can be branched or not branched, and that can be made of one single elongated cell or of multiple cells, such as here, that are combined with each other. But essentially, the point here is that they form an elongated duct that goes along the plant tissue. And so these epithelial cells here would now produce the compounds and would secrete them into the leticifer. And so this will be full of toxic kind of compounds. They can include rubber, the rubber trees using these leticifers. They can include lots of alkaloids, like shown here in the opium poppy, and so forth. But the principle is the same here. Like you have a way of secreting away the toxic compounds to prevent ototoxicity. And you now have a defensive structure where if insects are drilling into the tissue, that you have defenses coming out. Then last but not least, the root. Actually, that's more recent research, that the root has become more and more a point of research and a defensive point of actually accumulating these compounds. And so this is an interesting example from coleus. That actually is a Lamiaceae, so part of a plant family that more typically employs trichomes, and coleus does form trichomes. But it actually turns out that these type of defensive compounds here, in this case, it's a diterpenoid called forskolin, which also actually is an FDA-approved drug now for treating cardiovascular diseases, is actually accumulating in oil bodies here in the cell. So here we don't have sort of a specialized organ. We actually have these things accumulate in the root cells, but they're so hydrophobic that it's actually not as much of an issue because they're forming these oil bodies here. So you see every cell here has one oil body, really concentrating these defensive type compounds. There's still ongoing research about what the actual physiological functions of these compounds, and especially of forskolin is. So these are some major ones. There are obviously more, and we'll touch of them, right? There are some where we see they actually also just transported into the vacuole and stored there, not in specialized organs, but in suborganelles. We will look at some of these examples again when we touch on the individual metabolite classes. For now, I want to sort of move on and start giving us a bit of an overview of how these compounds are made. And you essentially can divide the biosynthesis of specialized metabolites, but really all bioactive plant metabolites, into two major parts. And the first one being the scaffold generation, and the second one being the tailoring reactions, so the functional modifications. And so what you can see here is, right, that this first step, the formation of the different carbon scaffolds, really taps into general metabolism now. So the origin of all these specialized metabolites comes from central general metabolism. And so these are your common pathways that any organism is using as a pool for metabolites for producing any kind of biomolecule, right? And so this is photosynthesis, obviously, glycolysis, glycolytic intermediates, or TCA cycle intermediates are really key metabolites that you can draw on to make more specialized compounds. And then specific to the nitrogen-containing compounds, the shikimate pathway is a key pathway. And then for the terpenoids and isoprenoids in general, the MEP pathway and the DOXP pathway are really critical central pathways to generate these kind of specialized metabolites. And then once we have made sort of this pool of metabolites, then really all sorts of functional modifications. The most common ones are glycosylation, methylation, acylation, prenylation. But the major one being oxidation, and some we also see reduction. So those are coming in, are really contributing to massively diversifying these base scaffolds now and adding bioactivity to these compounds. And again, we will look at these as we're moving through the different metabolite classes. Based on this chemical logic, the way that specialized metabolism has really exploded in terms of its evolutionary diversification and structural diversity is now duplicating and modifying genes that are doing especially these tailoring reactions. And so you're probably familiar with this concept, but I want to highlight it given how diverse our group is this year. If you think about, right, diversifying the scaffold forming functions, but especially the tailoring functions, is you want to have a pool of genes and a pool of enzymes to do that, right? And so the concept here, as how we envision that this evolutionary diversification has occurred, is that we would have, for example, a gene that is involved in general metabolism. And will actually, our first discussion paper is actually touching on how this may have occurred. So you would duplicate this gene through gene duplication or genome duplication. Now you have a second one that obviously can be under different selective pressure for further mutations. And so this is really kicking in now, because you now have two copies. One copy is not essential to survival anymore for general metabolism. So it can underlie further mutations and modifications, and then several of them. But there can be non-functionalization. We actually see this regularly in specialized metabolism, that you have a mutation that simply causes, at least temporarily, dead enzyme. You can have subfunctionalization or specialization. This is probably the most common ones, where you either have lost or gained additional functionalities, or you have now new substrate or product specificities or catalytic specificities in terms of mechanism occurring. So these would be falling under the neo-functionalization and specialization. But we can have conservation where nothing happens and you simply have two of the same copy boosting up that same pathway. Right? But so this duplication and the subsequent modification of these enzyme functions, whether they are catalytic in nature or whether you have now modifications in regulation, compartmentalization, and so forth, they really have contributed to this massive diversification and chemical range of specialized metabolites. Right? So we'll touch on this again as we move through the different metabolite classes. But this general concept is really important to keep in mind if we think about how these different metabolite classes and their biosynthetic pathways have evolved in individual plant families and species. And so just to sort of hone into this, how this actually looked like, to just give you an overview, the more hydrocarbon-based metabolite classes that we'll look at, they mostly come out of glycolysis. So you're actually using PAP and some other glycolytic intermediates, pyruvate as your end of glycolysis. Right? If this doesn't ring a bell, you might want to look back at your sort of base biochemistry classes. Right? Pyruvate can be modified to our central energy metabolite acetyl-CoA, and from there acetyl-CoA and pyruvate can, for example, go into the methyl erythroyl phosphate or the mevalonate pathways. These are the central pathways leading to terpenoids. Acetyl-CoA, right, is your key metabolite to make fatty acids. From those you can make the waxes. From those acetyl-CoA and mannonyl-CoA you can also make all the polyketides. So all of this is actually coming out largely out of the glycolytic intermediates and the TCA cycle intermediates. And then on the other side, we have the nitrogen-containing compounds. A lot of this is also coming out of glycolytic and TCA cycle intermediates. So the carbon backbones of your amino acids are coming from the TCA cycle, glycolysis, and actually your pentose phosphate pathway. Right? Especially through the PPP, you're getting the 4-carbon sugar erythrose. That is a key intermediate en route to the shikimate pathway. But key here is that you're using the PPP, glycolysis, and the TCA cycle to generate amino acids. And so out of those amino acids, that's actually what you're using now to make all your alkaloids, glucosinolates, cyanogenic glycosides. So these are your nitrogen-containing natural products coming directly out of the amino acids. And then we have the phenylpropanoids. They're an exception. They're not nitrogen-containing, right? So we have a special function that we'll look at, how we get rid of the nitrogen. Despite of that, they also come out of the aromatic amino acids, right? So all of your phenylpropanoids come through this pathway. So you can largely divide these two families of natural products by their biosynthetic origin. Right. All right. What we then see is, and we'll look at this again in the individual natural product classes, is to make these different scaffolds, there are a few sort of type of chemical reactions we need to look at. I won't go through all of them now. I just provided here as an overview of the type of reactions that we're actually looking at. We will probably not go into many of these specific enzyme mechanisms throughout the quarter unless you have a specific interest. But generally, to make these scaffolds, you're looking at, for example, amine aldehyde condensation type of reactions to generate nitrogen-containing cycles. Right? We find those, for example, in the alkaloids. Amine aldehyde condensations also intermolecular can happen. Again, oxidizing amino acids, we'll see in some compounds we're actually activating the nitrogen group here. Right? That's important. We'll see this later. Amino acid deaminations, we'll see. I just talked about the phenylpropanoids not containing nitrogen, so this might be a good way to go about this. So we'll look at that. Intermolecular cyclizations are very common, especially among the isopronets and the terpenoids. Acylation reactions in the polyketides are very common. Radical coupling also happens, but we won't really touch on it at all this quarter. These are very rare reactions that are not as relevant for our purposes here. But these are sort of the most common reaction types that I would encourage you, if they're really shaky on it, to maybe try and familiarize yourself with that a bit, that we use for scaffold formation. And then for our tailoring reactions, there are many others, but really the two major ones that are being used to tailor these scaffolds is oxidation. These are most commonly the initial reactions are oxidations of the hydrocarbon backbones. And then subsequent, we can see a lot of different group transfers, right? Again, methylation, acylation, prenylation, glycosylation, and so forth, are common group transfers that we'll see to modify the structure and the bioactivity of these compounds. And so looking at these modifications, there are a few sort of really central enzyme classes we need to look at this quarter. We'll touch on them again. The scaffold-forming enzymes are much more specific to these individual natural products, so we'll touch more on them when we actually talk about the individual metabolite classes. But the modifying enzyme classes are actually more generic, and so the same enzyme class actually is functioning in multiple groups of metabolites. And so probably the most widely distributed enzyme class that literally is involved in the production of every specialized metabolite class is the cytochrome P450s. So these are heme-containing proteins that can catalyze a vast variety of different modifications. Primarily, they are catalyzing hydroxylations and other oxygenations, but they can also catalyze dealkylations, dehydrations, desaturations, and so forth and so forth. An extremely diverse enzyme class, usually very large gene families, even within one plant species. So, for example, Arabidopsis, as essentially a very simple plant, has, I think, more than 400 P450s. So extremely diverse. These are gene families that have undergone extensive gene duplication and diversification. Their mechanism, however, is essentially the same, especially those that are catalyzing oxidation reactions. And so what they do is that they essentially use molecular oxygen and bring it into a molecule to hydroxylate or oxygenate a molecule under release of water. And they're actually to do that, they're using electrons that are coming from NADPH. So you do need an additional oxidation reaction that is coupled to the P450 function. And in this case, it is almost always NADPH. And so those electrons and that oxidation of NADPH is actually catalyzed by a cytochrome P450 dependent reductase. So these P450s actually always work in conjunction with one of these CPRs that is providing the electrons needed to facilitate the reduction reaction here using molecular oxygen. Right? So keep just this sort of very generic mechanism in mind. We will talk about a lot of P450s this quarter. I will not ask you to know the exact mechanisms, but I think it will be helpful for this quarter to understand what these enzymes generally doing. And so this is the most common reaction mechanism. Then for group transfer, I'm just sort of highlighting one group here. Again, we'll look into it again. These are the glycosyltransferases. The same would be generally true for other transferase type enzymes. Again, these are typically really large gene families, often functionally redundant, but also highly diverse. And so here highlighted just by example of the glycosyltransferases, they would, for example, take a hydrocarbon backbone that now has been oxygenated by a P450. So just assume this is a hydrocarbon backbone. You just hydroxylated this guy, and now the glycosyltransferase can come in and can use UDP-type glucose here and exchange the sugar moiety on the UDP and bring it onto the rest here And the OH group will go over. You will basically just leave this as an OH here, the donor that can take on another sugar moiety. But what you're doing is here you're using this UDP activated intermediate to get the sugar group onto your target molecule. And so this is just an example here against stereoside where exactly this has happened, right? You have a hydrocarbon backbone, you have hydroxylation, P450 mediated at all these positions here. And so those can now undergo the group transfer of glycosidation at these positions that's mediated by glycosytransferases. I'll show a little bit more of the mechanism when we actually get to some of these examples. But I think that's all I had for today, to just give you a first introduction into specialized metabolism. Again, I'm really sorry it didn't work out today to have a proper discussion. I hope we can resolve the technical issues, and that this video at least give you the material to work with and to learn. Please feel free to post questions directly to me or on Canvas, and we can continue the discussion there.